We are now live on YouTube, so if anyone misses out on the in the Zoom meet, they can definitely refer to the meeting on YouTube later on. And we'll be starting within just 5 minutes. Hello, everyone. A warm good morning, good afternoon, or good evening in whichever part of the world you are. I would like to welcome you all for the 2 days workshop on genomic data analysis. This workshop will help you get started with Genomic Data Analysis Program, which is an online bioinformatics training program that will provide an in-depth overview of concepts and analytical tools used to study genomic variation. The program is designed to introduce students, clinicians, and researchers to the role and applications of genomic data analysis to study genomic variation and apply genomics in translational sciences to improve diagnostic accuracy, predict which drugs are likely to be effective in patients, and contribute to the monitoring, treatment, and diseases in individuals and populations. Each week, participants will meet online to discuss various aspects of NGS data processing, genomic data analysis, and interpretation. After the first introductory lectures, participants will have the opportunity to get guidance and work on curated datasets or develop independent projects that leverage the tools and concepts covered in the program. In this 2 day workshop, we will discuss about genomic sequence alignment and data visualization, genetic variation and mutation, application of genomics in research, aspects of NGS data processing, genomic data analysis and interpretation, and introduction to sequence analysis in R and Python as well. I would now like to take this opportunity to introduce myself. My name is Sonalika Ray, and I'm the OmicsLogic Community Manager. I'm responsible for daily interaction with thousands of users that are a part of our community that is managed by Pine Biotech. So, before we start with today's workshop, I would like to introduce us for the ones who have joined this webinar for the very first time. We are a US based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy to use analytical tools, and our mission is to make bioinformatics more accessible. OmicsLogic is an international program that is running in 5 different regions with over 15,000 users around the world have completed the training programs. And due to this fast growth, our team is working with local and regional coordinators that are helping refine the local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. The programs follow a project based learning approach, which uses research grade tools to analyze data from top peer reviewed journals. OmicsLogic training has been completed by over 10,000 participants from 162 countries in over 300 workshops for 6 different specialization tracks, which include oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. Over the past several years, we have had the privilege of working with a number of organizations developing a training program for research oriented students, faculty, and democratizing bioinformatics, making this discipline accessible to thousands of students and faculty around the world. I'm honored to introduce the founders and team members of the company. Dr. Alfred Tauber comes from a medical background with an expertise in oncology, immunology, and biochemistry of inflammation. And one of his major projects is the establishment of Tauber Bioinformatics Research Center, which has been at the core of what we do. Dr. Leonid Brodsky is the director of Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. Our platform is a result of hard work by some of the people mentioned here on the right. Julia Panoff is an expert bioinformatician. Avi Titovsky leads the development of the T-Bioinfo platform. Dr. Harpreet has an expertise in the application of statistical machine learning techniques on biomedical data and looks after the curriculum development, bioinformatics, and data science course development. Dr. Mohit Mazumdar is an experienced bioinformatician with multiple publications in peer-reviewed journals. He has been developing collaborations with universities as a mentor and project lead for OmicsLogic training and research fellowship. Dr. Raghavendran Lakshminarayanan is an accomplished computational biologist with an experience in large-scale analysis of multi-omics data and data science methods in precision oncology and protein DNA structural bioinformatics. So that was all about our company and our team members. Now, to get started with today's workshop, the first very important thing is to sign up on the learn.omicslogic portal so that we can get started and also go along with the mentors to follow the pipelines on the server. I just had a text in the chat box that I wasn't audible. Was it for anyone else as well, or was it just for Nadia? Oh, just
So
can you please
Sonalika, it
confirm?
was
It's fine. It's fine for us.
Okay, Nadia,
yeah,
can you
it's fine.
please try rejoining then? Try to rejoin the same link. Thank you, Jasvi, for sharing with us the link to the portal. So for those who have already created an account on the portal, they can proceed further with updating their profile. But those who are new to the workshop and also are new to this platform, it would be nice if you just could quickly create your account on the portal so that when mentors take you through the resources and the server pipelines as well, you are able to go along with the mentors. So I'll just give you a demo of how you can sign up on the portal very quickly so that you can follow the very same steps. So you just need to click on the login button that is on the extreme right top, and you will be then redirected to the sign up button. Now here, either you just put your mail ID and password, like create an account for yourself and sign in, or you can also sign up using any of your social media accounts that could include your Google accounts, Facebook, LinkedIn, etc. So I'll just log in quickly with my Google account. And those who have already signed up can please let me know in the chat box with one or yes, so that I know that they are done signing up. And others, if you're connected with your desktop or laptop, then please sign up now and let us know in the chat box so that we can proceed to the next phase. Thank you, Rajeshri. Okay, now let me just also sign up. Thank you, Samriddhi. Now, for those who have signed up on the portal, it is very important to have a complete profile. Now, what is a complete profile? So it's very important that you should add a profile picture to your account. Your name should be in title case. That is, the first alphabet is capital, others are small. It should not be capitalized. It should not be in small letters, because this is the same format that will reflect on your certificate. Also, connect your social media accounts and add a brief bio about yourself that could include your educational background and your research interest. Now, for every coursework that you will complete, you will gain some points that will take you from a beginner level to an advanced or an expert level. And you'll be able to monitor your progress, that is, the coursework that you have been taking or have completed, the certificates that you will get after completion of each coursework here in your profile tab itself. So it's very easy to download a certificate after the completion of a particular coursework from your profile. So when you click and complete all the lessons of particular coursework, we'll shortly take you through the coursework and show how they are arranged. You can simply download your certificate by viewing it from here and share your achievements on social media platforms for the mentors to recommend you. And just to let you know how you could navigate through the portal, so here is the courses icon in the menu bar on the portal itself. Just click on courses, and on the right side, you'll be able to find a list of tags. Now, these tags refer to the various courses on multi-omics and data science. The courses on multi-omics include transcriptomics, genomics, metagenomics, epigenomics. Here is the list of all the courses. So you can simply pick the tag that you would like to explore more and get the list of courses or projects that you would like to learn more about. So that was the very simple way how you can navigate on the learn.omicslogic portal. And before we get started with the workshop, it's also very important to make sure that you have your account working on the tbioinfo server. So for those who have joined any of our webinars for the first time or have just signed up on the portal, I'll be giving you or sharing with you all the demo account credentials, and you can definitely refer to these credentials to sign up on the portal and run the pipeline that our mentors will be taking you through today. And those who have already signed up on the portal, they can use their same OmicsLogic Learn credentials to sign up on the server tbioinfo, or they can also, those who are program participants or are the participants of research fellowship program, they might be having their customized credentials, so use that. So that was all that I wanted to share before we get started with the workshop. Now I would like to invite our mentor, Dr. Raghavendran, to please let us get started with the genomics workshop. Over to you, sir.
Thank you. Thank you very much, Sonalika. Am I audible?
Yes, sir.
Yes. Thank you very much. Let me find methods to share my screen. Can you see my screen?
Yes, Dr. Raghavendran, we
Thank
can.
you. Yeah. Okay. A very warm welcome to all of you to this session. So today we are going to discuss genomics, what is genomics data, how it is organized, and how it is interpreted to be such a powerful resource in the hands of clinicians, researchers, and entrepreneurs, and how reading the DNA code will help us battle cancer, will help us fight off infectious diseases, and will help us even solve some crimes. So let's start by reviewing the fundamentals of genomics. So genomics is all about DNA. So let's review what is DNA code. So what do we already know about this, and what kind of answers can we find here? How can we analyze huge sequences of nucleotides stored in the DNA, and how do we make sense of them? Just like a book, the DNA code is organized as letters that form sentences, and sentences that form paragraphs, and paragraphs that form that are organized into chapters. Knowing how to read this code can help us decipher the full story about who we are, and to look into our history and to understand how we are connected to other living things around us. So DNA is short for deoxyribonucleic acid. So it's a long molecule composed of two chains that coil around each other to form a double helix. This double helix carries genetic instruction for development, for function, for growth and reproduction of all known life. So this all known life includes multi and single cell organisms, and as well as many bacteria and even some viruses. So yeah. So the DNA code contains directions or recipes for life, including the capacity to grow, including the capacity to reproduce, and including functional activity that characterizes the cells, and including the continuous change that proceeds until the cell dies off. Since life is a condition that distinguishes animals and plants from inorganic matter, understanding this DNA code is synonymous to understanding life itself. So the instruction for life of a single cell, and also a life of a complex organisms like us, humans, which contains over 37 trillion cells, right? Each cell contains the same DNA, which is stored in a nucleus. When cells divide, as they do when new organisms is formed, or when new cells has to be generated, we see how this DNA is organized. The nucleus is opened up, and inside we discover these tightly packed strings that can be visible under a microscope. These are chromosomes. Chromosomes contains different genes. Each one of our cells contain 23 pairs of chromosomes. One of the chromosomes come from one half of the chromosomes come from our mother. The other half comes from our father. Yes. So this unique structure of chromosomes keeps DNA tightly wrapped around in a spool, wrapped around proteins like a spool, and these proteins are histones. So without such packaging into tightly wound chromosomes, DNA molecules would be extremely long to fit inside the cell. For example, if all of the DNA molecules inside a single human cell were to be unwound from their histones and to be placed as end-to-end as like a single string, that would stretch to reach to 6 feet. So the largest chromosome of all the chromosomes is chromosome 1, which contains about 8,000 genes. And the smallest chromosome is chromosome 21, which contains about 300 genes. So once the cell division is complete, the information on the cell that is stored in the DNA helps the new cell know what to do to continue its function that it will work on for the rest of its life, and preserve the DNA that it has been passed on from its parents and to pass on to the next cell. And in the meantime, the DNA that contains instruction for various functions in a cell must perform to grow and maintain life and function. Therefore, the DNA contains both the necessary ingredients like all the recipes that is required for the cell to function, and all the other instructions that is required for the DNA to be stored and accessed at the specific time. Together, all of the specific code of chromosomes that we are dealing with or that we are discussing with is called a genotype. The product of this code and how it interacts with the environment is called a phenotype. A phenotype can describe a single cell, a tissue, an organ, or even the whole organism. A phenotype of an organism could include height, size, weight, or activity. And phenotype of a cell could include its specific function, its size, its shape or morphology, and its composition. So DNA is made up of nucleotides at the basic level, at the base. It's a fundamental level. The DNA is made up of nucleotides. A nucleotide is made up of a sugar called deoxyribose and a nitrogen containing base and a phosphate group. So the nucleotides are joined to one another in a chain by covalent bond that forms between sugar of one nucleotide and the phosphate of the next, resulting in an alternative sugar-phosphate backbone. So each strand of a DNA molecule consists of a sequence of bases that is complementary to the bases in the opposing strand. So the pairing between these complementary bases is what gives rise to the characteristic double helix shape. So in this structure, adenine is bound to thymine and guanine is bound to cytosine. So to understand how DNA works and to read its sequence, a special project was aimed at decoding DNA that was launched in 1990. So this 13-year-long project was initiated in 1990 and is focused on determining the DNA sequence of the entire human genome. So scientists set out to determine the DNA sequence and the location of estimated 100,000 human genes at the time of the initiation of this project. Many at that time believed that once we had the sequence, we would quickly be able to determine where all the genes were. And this process of reading the DNA sequences and piecing them together laid the foundation of what we are doing today, laid the foundation for genomic study and the foundation for all the other omics study that is thriving today. Yeah. So, but simply reading the code and annotating it became a much longer process than sequencing the genome itself, than the Human Genome Project itself. Today, there are several different competing human gene databases with many thousands of differences among them. Although the number of protein coding genes has gradually converged and the number of other gene types has exploded. So the latest count has consistently has found over 22,000 genes long since the Human Genome Project was completed. So we have around 22,000 genes, protein coding genes, and that forms only 1 to 2% of content that we have in our whole genome. So in brief, the DNA is stored in the nucleus and it is organized into chromosomes and into specific regions. And some of these regions are easier to read and to access and to be evaluated for further functions. And these easy to read regions are called protein coding genes. And protein coding genes are the sequences, or the genes or sequences that is used to produce proteins. So how it is done? The central dogma of molecular biology describes how this is done. This is done involving a two-step process, transcription and translation, by which the information from the gene is converted first into RNA and then to protein. The information flows from genes to proteins through DNA being converted to RNA and RNA being translated to protein. So in each step of this transformation, we see how the DNA code is used. First, for transcribing the source code to a working repository of RNA, where some code is modified and some code is not used at all, and then it is released for producing a functional product called protein. So this process has facilitated several key proteins, one of which is facilitated by several key proteins. So one of which is called DNA polymerase. This DNA polymerase is the one which attaches to the DNA as it unzips to replicate the DNA to form single strand to single strand, and then it adds complementary bases to the single strand and creating a second workable copy. This copy DNA code can in principle be used in other cells also, but yeah, it is can be used in other cells also, but it is also important to make sure such a replication process goes without any mistake, so that the cell doesn't end up with a different set of copied DNA than to the original set. And the cell also has a lot of mechanisms and other products and instrumentation in protein complexes that are designed to prevent this error or in error correction mechanisms also. So each gene, as we saw, is a recipe for certain protein. So the gene sequences encodes instruction from which a protein can be made, and the DNA sequences encodes the instruction from which a protein can be made. So proteins are the products of amino acids, which are encoded by the three sequences or trinucleotide sequences that are called codons that are stored in the DNA. So the DNA sequences they are read by the jump of three. First, three sequences are read and then converted into one amino acid, and then goes the next three, and then goes the next three. So a codon is a sequence of three nucleotides that corresponds to specific amino acid or a stop signal during a protein synthesis process. Once a right place on DNA is copied over, it is prepared for translation by forming a messenger RNA that is exported from the nucleus. So the translation happens in the cytosol, not in the nucleus. So in humans, we saw genes are very important and they make up protein. And in humans, genes vary in sizes from few hundred DNA bases to more than 2 million DNA bases or 2 million nucleotides. The keratin-associated protein, for example, is 447 nucleotide long, but whereas CNTNAP2 gene has something like more than 2 million nucleotides. So the number of genes also varies between various organisms. For example, the influenza virus has only 11 genes, while the grape genome has over 30,000 genes. So the information is not uniform across the different organisms. Neither the information about coding proteins is uniform across the genome itself. So the genes are not just sequences of nucleotides. In fact, they have regulatory sequences at both the ends of the gene, that is upstream and also downstream. This regulatory sequences controls the level of transcription of each gene. So inside the gene sequence itself, there are protein coding regions called codons or exons and non-coding regions called introns. The cell's machinery perform a process called splicing that will leave out or that will exclude the introns and then include only the exons before the gene is translated into proteins. And the extraordinary length of some of the genes has enabled the presence of introns, and this presence of introns and multiple introns provides a mechanism for generation of transcriptional diversity through alternative combinations of different exons at different levels. So the translation in the cell is done by ribosome, which can read the mRNA sequence and assemble the protein, and this protein can then fold into a functional form or functional structure. So can you see my screen? Yeah, I think yes, now it is working. Yeah. So when we want to understand the function of a particular protein, we typically combine the information about its chromosome position, about the coding sequences that it has, about the regulatory region, about the structure as well as functional features of the protein, and about all these information which are understood through thorough experimental validation and or its comparison to proteins from the same family or proteins that has the same structure. So now we understood the context of what is DNA, how it is organized, and how it is used, and how it is translated to form protein, which are the machinery that defines the characteristics or that defines the phenotype of the cell or that defines the functions of the cell. So let's discuss how to analyze this DNA code. Let's look at the sequence alignment and identification of variant that have functional consequences on the phenotype of the biological system currently under study. So let's go back to a little bit of a history. So the year 2020 marks a 70-year mark since the discovery of structure and function of DNA molecules. It took almost 50 years from the time to sequence the first human genome, but soon after, the technology evolved and exploded rapidly. And by 2015, we have the 1000 Genome Project, which brought a significant number of whole genome sequences to the research community. And in 2018, we have over 100,000 whole genomes that were sequenced. And today, this is an ever-increasing number of, there is an ever-increasing number of different omics data sets that are available that will describe cellular and subcellular process in a detail that cannot be imagined something like 50 years ago. So the omics, the multiple omics that we just that you just heard, aims at a collective characterization and quantification of pools of biological molecules that translates into structure, function, and dynamics of these organisms or multiple organisms. The omic technologies are used to explore the roles, relationships, and actions of various types of molecules that make up the cells of an organism. So many types of omics data that can be generated using high-throughput sequencing or next-gen sequencing can show detailed information about genomic variants, epigenomic regulation, phenomic data types, and the different transcriptomic states of the cells that we have in our study. The suffix "om," one used to define various fields, actually originated from in the 20th century. In the 20th century, when genome was first coined in Germany in the beginning of in the 20th century. The word genomics refers to the totality of the genomes, or the study of all the genes and genomes that is present in the cell. A genome is an organism's complete set of DNA, including all of its genes, as I mentioned, in contrast to genetics, which specifically refers to study of individual genes and their role in inheritance and their characteristics that defined from this inheritance. Genomics aims at a collective characterization and quantification of all the organism's genes, their interrelations, and influence on the organisms. As a result of such effort, we can now detect differences which can be harmless or differences that will lead to serious issue when a gene is produced. So just like any code, we can expect the DNA code to contain mistakes. So these mistakes occur as a result of copying the DNA source too many times, as a result of various stresses that can interfere with the with this copying mechanism that is inside the cell. So to understand how genomics is used, we need to understand the analysis approaches to DNA code, specifically how we can identify the changes or how we can identify the mutations that characterize them, and to interpret the significance of these changes that we naturally observe, or that we observe when we sequence the DNA. So mutation can actually happen in several different ways. So one form is substitution. A substitution mutation is something that can cause several consequences to DNA code, but it implies a minimal change that only leads to a single nucleotide change. So when one nucleotide is substituted with the other nucleotide, it's a straightforward substitution. And insertion, however, changes the number of DNA bases in a gene by adding a piece of DNA. As a result, the protein that is made by the gene may not function properly because the reading sequences have completely changed due to the insertion of this one or multiple DNA bases. A deletion mutation, however, changes the number by removing one or certain pieces of DNA. Small deletions may remove one or few bases within a gene, while larger deletions can involve removing an entire gene or an entire exon of several neighboring genes. Deleted DNA sequence may alter the function completely alter the function of the resulting protein, and that is going to interfere with the cell's processes. So a duplication mutation or duplication event consists of a piece of DNA that is abnormally copied one or more times. This type of mutation also alters the function of the resulting proteins, and which will also interfere with the cell's machinery. So a specific type of variation can lead to harmful results. For example, a change in function is harmful, and this is called mutation. Or in the other way, when the changes in these variation is not harmful, then that will not be that is not a that's a neutral mutation, or that is no mutation. That is a variation. So for example, in this case, variation causes the eye color to change, while mutation causes some really horrible deformities and functional losses. So when these genomic variants are discussed, both mutation and both the term mutation and polymorphisms are used interchangeably, but by convention, in human genetic research, any genomic variant with the population frequency of less than 1% is termed as mutation, while any genomic variant with the frequency of greater than 1% is termed as polymorphism. So this distinction based on the population frequency of the variant rather than the type of the variant is based on the variant or the type rather than the type of variant or its possible functional impact. However, as a rule, rare mutations that are rare, that is even less than 1% observations, tend to have functional impact that deviates from the wild type, yield to higher signal to noise ratio for detection of a genetic influence of a trait, or a rare mutation that will have functional impact can have very drastic effects in the influence of the trait. In contrast, polymorphism that is observed tend to result in less functionally deviant genomic variants. It might not cause severe deformities or severe functional impact of the cells or of the organisms in study. So, however, it is also worthwhile to think, or it is also worthwhile to keep in mind that these rules, there are exceptions to these rules. It is also observed that polymorphisms can be, some of the specific polymorphisms can involve loss of function, and some of the simple variations in gene mutation can lead to no loss in function at all. So, a single nucleotide mutations can be of several kinds. So this is a single nucleotide mutations. Substitution mutations can be of several kinds. One of this kind is missense mutation, which changes only one DNA base pair, and that results in a substitution of one amino acid for the other in the protein made by the gene. A nonsense mutation, however, changes one DNA base pair that prematurely causes the cell to stop building the protein, or it introduces a stop codon in the DNA sequence that prematurely stops the length of the protein that is being synthesized. And this type of mutation results in a shortened protein which will not or which might not function properly at all. There is another type of mutation which is a frame shift mutation, where an addition or a deletion of DNA bases completely changes the frame of the gene's reading. As I had introduced you before, the codons are read 3 bases once. So when you delete or insert one or few bases, this reading frame changes, and by changing this reading frame shift mutation occurs, and this will result in a protein that is completely different from the original or completely different from the wild type protein, and this could usually result in a non-functional protein. So to summarize these different genomic variations can be of several different types. So when analyzing the genomic variants or genomic data, we are looking to find and characterize these changes in the DNA code and associate such changes that we are observing to the phenotype or to the different cells characteristics. Here are some of the acronyms and some of the words and jargons that you have to remember about genomic variations: SNPs and SNVs. So SNV is a single nucleotide variant, where a single nucleotide change that may or may not be present in the population. Basically, this means any change from the reference genome that we find in a given DNA sequence above the level of technical error. So SNP is on the other hand, is a single nucleotide polymorphism, where a nucleotide is known to be altered in a proportion of population and typically considered to be prevalent in approximately more than 1% of the population. So this is a single nucleotide polymorphism is a mechanism that causes blue eyes and green eyes and different eye colors in the in different human races. So another form of acronym that you should remember is copy number variation, where the same gene is encoded, or same gene is being copied multiple times and is present in different number of copies across different cell types or across different conditions of the cells that is under study. So such changes as well as the characteristics of these different variants are commonly referred to as genotype of the cell or genotype of the organisms. So this is what we are studying by understanding or by sequencing the DNA sequence, and by trying to analyze the DNA sequence, we are trying to understand the genotype of the organisms, or we are trying to understand the genotype of the cells. So the bioinformatics technology that will allow you to generate the digital information from these biological samples is called Nextgen Sequencing or high throughput sequencing. This technology was first developed during the human genome project, as discussed ago, and has evolved tremendously since then. So over the years, many different sequencing techniques were introduced, and they were developed and perfected to study whole genome sequences of multiple organisms. So sequencing platforms use variety of read lengths. So read lengths is the length of the DNA sequence that can be read at once, and this varies by different sequencing platforms. But in general, they can be divided into short read sequencers and long read sequencers. So the benefit of the short read sequences isn't that because they are short, but they can be extremely inexpensive, and they can be generated in a massively parallel manner with billions and billions of reads being generated in a single run. So the cost, when the cost needs to be kept down, and when you have to sequence multiple different samples, and you have to perform it a lot of times, and the read count has to be really high, and then short read technologies makes most sense. While long reads, on the other hand, can resolve regions of genome that are inaccessible or that are inherently not possible to assemble due to the length of the short reads, because these regions can involve repeat sequences, and those repeat sequences may interrupt our read mapping process. So in that case, long read sequences have advantages over short read sequences while dealing specifically with those regions which have repeat sequences. And latest advancements and recent advancements made by Oxford Nanopore Technologies, for example, under right conditions, under perfect conditions, under perfect high quality DNA, can even produce reads up to 1 million bases. That is huge. So this technology allows researchers to explore the genetic variations like never before, down to a single nucleotide resolution. Whether you need to identify specific variants that from a focused set of regions, or to look more broadly about potential different variations or causative variations, or to look for differences or variations in the number of variations in the number of times a gene is being copied. So these are all different explorations that one can perform. So based on this, whole exome sequencing or whole genome sequencing can both offer effective solutions based on your question to what to look for and how to design your data. Whole exome sequencing or whole genome sequencing, as I mentioned, can be both effective. So whole exome sequencing is a targeted method to sequence all protein coding regions in the genome. It offers reliable and sensitive detections of coding variants, and it is fast and it is cost effective, as it sequences only fraction of what the whole genome sequencing is going to perform. Whole genome sequencing, on the other hand, provides a comprehensive view of the genome, including coding, non-coding, mitochondrial DNA, and everything that is present in the cell. So it is reliable and sensitive as well, but includes the process is complicated, and it also it can provide you with the necessary information about detection of different variant types that is under study or that is under question. And these variant types or these variations and mutations, these variations are not limited to coding regions alone, because we are sequencing the whole genome. So now let's understand about how this process is being employed and how this is being done in a very short intro. So the NGS technology that captures sequences the DNA bases actually captures and generates this digital data using several steps. Here, the whole genomic DNA is isolated, and then it is sheared, and it is shattered and sheared and placed in a flow cell. So inside the flow cell, the DNA fragments are amplified using bridge PCR amplification, and then the flow cell is inserted into the sequencer. The sequencer machine uses image analysis to capture each and every nucleotide in this flow cell fragments by analyzing the visual patterns and converting them into sequence of letters, that is TCGA, that are the nucleotides that forms the bases. Eventually, the sequencer will produce reads typically of 30 to 300 base pairs, base pairs in length. So this sequencing data that is emerging or that is coming out of the sequencer is stored in several different file formats. So for example, a Fastq file format will provide us reads with both will provide us the reads from the sequencer, and it is text-based file format. This stores the nucleotide sequences of reads and the quality scores for each letter that is sequenced. The FASTA format, on the other hand, stores the representing DNA and protein sequences, and this is typically reserved for the reference file of the genome or reference file for the genome to which the reads that are in Fastq files are matched to. Then we have the genome transfer format, which contains the genome annotation. For example, a genome transfer format, GTF file, will store the start and end of each genomic element like exon, like isoform, or like gene, and other sequences, other elements that you find in the whole genome sequence. So, can somebody mute their microphone, please? Yeah. So in short, a FASTA file contains a genomic sequence itself with a line that's starting as with the line that starts with the greater than symbol, which is a header that describes a sequence, and this is and describes the sequence. For example, this header describes a species from which the sequence is for which the DNA sequence belongs to. In this case, it's from a virus, and in this case, it's from a Frugiperda fungus. So a Fastq file, on the other hand, is also a text-based format, but it stored the sequence, and it also stores the corresponding quality scores of each and every letter that is being sequenced. So both the sequence letter quality scores of each RE are encoded in a single ASCII character. So a Fastq file normally includes information about the name of the read, its length, the raw sequence letters, and the quality values for each sequence for each nucleotide. Depending upon the sequencing method that has been employed, Fastq files can be quite large, sometimes over several gigabytes in size per file. So to analyze such raw data, many different tools for genomic data analysis are used. These analysis tools include alignment of reads and full sequences, analysis of relationship between sequence and their evolutionary divergence, and measurements of gene expression, and relationship between sequences and structures, as well as relationship between somatic and germline variant calling methods, and multiple different techniques that will characterize somatic and germline variants. So the platform, the tBioInfo platform, the tBioInfo is a big data platform that can be used to perform all of these analysis and many other using an intuitively user-friendly interface. So the sequence alignment that I mentioned ago can refer to two types of alignment. The sequence alignment to a reference, in which case it is called mapping. So where you have the Fastq files, and these files are from the sample, and you wish to align these files or reads to its best position on the genome to find whether the sequence in the read or multiple reads reveal variation from the reference which are in FASTA format. So in other type of alignment, which is multiple sequence alignment, this is what you do when you have multiple consensus sequences that you would like to compare each one of these sequences for their differences or for their similarities, and develop an understanding between them in terms of how these sequences or portions of these sequences are conserved or portions of the sequences are varied. So in many cases, these multiple sequence alignments are used to find structurally important variation, and it can also be structurally important variation in both nucleotides and also in protein sequences. So when we have the Fastq files ready from the sequencer, and when they are ready to be analyzed, there are several important steps to be considered for performing the analysis. These steps include pre-processing, mapping, variant calling, and annotations. So for each task, you can connect analysis jobs to a pipeline. Each one of these various inputs, a pipeline will have various inputs, and each of these algorithms in this pipeline will have multiple outputs, and a pipeline is defined by joining several algorithms together, from where the output of one algorithm straight away or directly feeds to the input of the other algorithm. And in tBioInfo platform, the outputs at every step is presented to you and is given to you, and you can analyze or you can visualize where the information is changed and how the information content is built upon, and how the information content is maximized at each step of the at each analysis step of this pipeline. So however, to carry out a genome-wide analysis study, for example, researchers use two groups of participants, let's say people with the database, people with diseases being studied, or people with without diseases. Sometimes this genome-wide association studies can also be performed to identify certain variations of disease or conditions that we have in hand. So how is this performed is when the researchers obtained DNA from each participant and sequenced their whole genome, and if a certain genetic variations are found to be significantly more frequent in people with the disease compared to people without the disease, then these variations are said to be associated with the disease. So identifying these associated genetic variations can serve as a powerful pointers to understanding those regions of human genome where the disease causing problem resides. So the methods that I just mentioned before downstream analysis or visualization and somatic mutations can help us understand and can help us relate these genetic variations to these different regions which might be involved in causing diseases or causing problems. So yeah, so in this case, it's easy to understand, or it's easy to observe that chromosome 14 has a higher mutational rate that could be involved in the disease causing state. So downstream analysis will actually identify mutations, SNP variations, and these mutations and SNP variations can be presented in multiple different ways depending upon the purpose of analysis. And in this case, it can be presented in a tabular format, or it can be presented in a simple graphical formats depending upon depending upon the kind of report that you are generating based on the analysis that you just perform. So this program is actually designed to help you learn more about these analysis methods and their applications in various project examples. So this genomics program will include an expanded overview of key concepts, principles, and examples of genomic data analysis. Now I want to invite Dr. Harpreet to present you with a couple of demonstrations of such analysis which can be performed in T-BioInfo Big Data platform. So over to you, Dr. Harpreet, and then we will come back for the for how to briefly talk about how such genomic studies can be used in real life. Can I hand over the podium to Dr. Harpreet for a demonstration?
Thank you, Dr. Raghavinder. Thank you for a very nice session. So already Dr. Raghavinder was talking about like what are the different mutations, how you can analyze that, how we can perform phylogenetic analysis, what is the advantage of such kind of analysis. Now let's see how we can do that practically, particularly by using a T-BioInfo platform. So now let me just give me a second. So just let's go over the server. So my screen is visible to everyone. So this is the T-BioInfo server, which actually has multiple sections where you can perform various type of analysis depending upon the data that you have, then depending upon what type of analysis that you want to perform. So this is very important platform. This is especially for both people who do not have any bioinformatics background, as well as for those people who already have bioinformatics background. But as we know that NGS data has very large data and it is impossible to analyze such kind of data on your own system. So this platform offer you the opportunity to give this interface to perform various types of analysis without, even if you don't have much knowledge about programming, because here scripts are already executed. So then you just have to perform like a click on button steps. So you just need to understand the logic. So for example, here under the NGS data, you will find various sections like transcriptomics, that is bulk RNA transcriptomics, then genomics section. Then we also have a section that are associated with microRNA or repetitive elements under the DNA and RNA. Similarly, we have some of specific mutational analysis section under the cancer biology. Then as we know that these days single cell data analysis is like nowadays it is so you will find specific section for single cell data analysis. Similarly, for virology, metagenomics, and data mining. So now, as we are talking about genomics, so one of the aspect that so far Dr. Raghavinder was talking, that is phylogenetic, how we can perform multiple sequence alignment. So let's first go over one of the sections that is under data integration and modeling. Here you will find this data association, and under the data association subsection, you will find this section specifically dedicated for phylogenetic analysis. So here, when you find this section, that is means already example pipeline or demo pipelines are already given there. So what we mean by demo pipeline, it means you need not to upload any data. So we already loaded the data. You will understand what are the steps that we can perform and how we can interpret those results. For example, let's take this example, that is demo wastewater SARS-CoV-2 MSA, that is multiple sequence alignment. So this pipeline where we are comparing the samples that were taken from particular SARS-CoV-2 patients and SARS-CoV-2 infected patients, not a patient. So here they have already collected the wastewater data, particularly from the UK, where they have samples particularly available in the wastewater itself. So we are going to this demo. So by clicking on this, we just able to upload data automatically, where we already uploaded raw sequencing files that were obtained from wastewater samples. So then why start? We will able to upload the data itself. So as Dr. Raghav was talking about Fastq files, so they are the files that obtain directly from the sequencer machine. So it contains the reads. So then once you have the reads, next is how you are going to align your reads on reference genome. So for example, here we were talking about SARS-CoV-2. So we were using reference genome for SARS-CoV-2, and with this algorithm, that is BOTI2. So this algorithm will align your reads on reference genome. So this is one of the mapping algorithms. So then the next step, which is automatically get highlighted, that is to create the consensus. So here you will find first option which is getting highlighted, that is per nucleotide. So what is per nucleotide module? So it will create this kind of table where you have sequence, then corresponding ATGC positions. So then once you have those positions, then you will able to create the consensus sequences. So once you have the consensus sequences, then we can perform multiple sequence alignment that will align our consensus sequences that were obtained from different samples. So it automatically align all those algorithms, all those sequences. So specifically here the algorithm that perform multiple sequence alignment, that is omega W. So then once at the nucleotide label, you are able to align your sequences. So then next is, because to study the phylogenetic analysis, we also need to convert a nucleotide sequence into the amino acid. So here this particular module will convert, or because on the reference genome we already provided translational codons. So translational, with the help of translational codon, so your nucleotide multiple sequence alignment sequences will be converted into the amino acid. Next is you're just selecting the codon position. So then so after you're able to perform multiple sequence alignment successfully, next is how you can generate phylogenetic tree. So you will find various algorithms here itself, like ape, and then you have algorithm like beast, coalescent, beast speciation, you'll process, beast birth death process. So then particularly most of the beast algorithms not just consider the similarity, they also consider the time points. So you can here we are using this beast patient birth death process algorithm to perform phylogenetic analysis or to generate a phylogenetic tree. So then once we able to perform the steps, next step which is getting highlighted here, that is end. So once we able to end the pipeline, so we will able to direct it to this interface where you will able to get all the output files. Since this is already an example pipeline, so we are immediately able to get the results. But yeah, in case when you are doing all this analysis on your sample, usually it takes sometimes depending on how many samples you are uploading, so it can take 1 or 2 days if you have very large number of samples. If you are just using very few number of samples, in that case it can take maybe 1 hour or something like that. Now let's look at what kind of outputs that we will obtain. So let me first show you, for example, simple phylogenetic tree. So here you will find even a PDF file. This is the PDF file. For example, now you have this kind of phylogenetic tree where you can compare which sequences based on the phylogenetic tree. You can understand which sequences are more similar to each other or which samples are more similar to each other. So the sequences or the samples that are present in the same clade, it means they are more similar. For example, if I'm comparing these two, for example, these two samples, they are more similar in comparison to if we are considering samples that are present in different clades. Apart from that, if you have already time point information, in that case, the another very important information that we will get, that is like which particular sample originated first and what are the other sequences. For example, here in a phylogenetic tree, how you can understand. So if you have any sample that are towards the root, so here, so those are the old samples in comparison to if you have the samples that are in the nodes. So if this length is larger, specifically, for example, these samples might be the new one. So then you can compare, if you have the date, even the time information, then you can understand which sequences are originated first or which are originated later. So you can compare samples based on the phylogenetic tree just to understand which are the evolutionary, specifically phylogenetic tree help us to understand what is the evolutionary relationship between different sequences. For example, here we have reference genome, and this is also, we just taken another reference genome sequence just to confirm whether the samples, if they are present in the same clade, it represent actually more similarity. So for this, what we did, we taken the reference genome, then we just taken another reference genome, we just name it as outgroup sample. So then when we are looking at these two samples, so they are present in the same clade. It also show or confirm us like these sequences are more similar to each other. Now let's look at the next very important output. So this is like phylogenetic tree. Now how we can visualize or understand these results, particularly at the level of multiple sequence alignment, right? So these are the like final results. You cannot compare what is the differences between these. So right now you know that these sequences are, for example, these two sequences are more similar to each other in comparison to other sequences. So how do we understand what are the positions which are different from each other? So what kind of analysis or from which type of analysis we can understand that? Now let's go back to our outputs. So first output that you will obtain here. So if you look at here, so I'm just reading its name, that is ends toward, it is MSC out underscore FASTA.fa. So this is multiple sequence alignment file at nucleotide level. Now let's download this file, and now let me visualize this file using a freely available tool, that is UGene. So now let me open the UGene. So this is the sample that we downloaded right now. So let's open this here. Now here we have multiple sequence alignment at a nucleotide level. So this is what multiple sequence alignment, which we can visualize using UGene. Now how we can identify where are different sequences, or all these sequences are mutated, or where there is a mutation. For example, whether you are considering with respect to reference genome or you considering a specific sequence or a sample. So how we can find out what are those mutations? So on the gene, you will find this option that is looks like a graph. So if you click on this, so here we need to select first that reference sequence, whether you want to compare your data with respect to reference or whether you want to compare your data, for example, with a specific sample. If you have, like, you are considering control samples, right? So I am comparing my data with respect to the reference genome. So this is also my reference genome, and I'm just scrolling these samples in this way. So first I have to select a reference genome. If you select it here, just select this sequence, then go to the reference sequence. So here you're able to select this. Now, how we can identify how many mutations are there? So we can find number of mutations as well as percentage of a mutation. Let's look at first simply number of mutations. So here you can select this option, show distance column. Then you can go to the count. So here you will find this column, score column. So these are the sequence name and or sample names. Then you have this score column that will let you know how many mutations are there in comparison to the reference genome. This is a reference genome. So how we can compare it? So we can look at, for example, this is the same sequence. We just named it different. These are both the reference genomes. So there are 0 mutations. When you're comparing, this is the sample 1. There are 7 mutations. This is sample 2. We have 10 mutations. Similarly, we have sample 8, 10, 7. Here we have 6 mutations. So this is how we can look at how many mutations are there. Now, where are those mutations? We can always check that as well. So if you just scroll this, particularly you start scrolling this to left to right, you will also able to identify the mutations. For example, here. So for this, at this particular position, that is 9634. So you will find this A is getting mutated to the T. If you know that which particular protein have the sequence start position, end position, you can definitely identify what are these mutations. So this is how we can look at what we can interpret from the phylogenetic tree, then how we can and what we can interpret from the multiple sequence alignment itself. Is it clear to everyone? Or if anyone have any questions so far? Okay, Daniel, anyone else? If you have any specific question, please feel free to ask your question before I move ahead. Okay, so if there is no more question, let's go back to the server again. So this is just one example, like if you have the sequences, you can do the phylogenetic analysis to understand what is the origin or what is the old sequences, how similar they are. So then what are the different where and what are the positions where mutations are taking place? You can perform those kind of analysis using MSA. So then what other options that we have? For example, we have, this is with respect to the viruses. Then we have specific, for example, when we are comparing cancer and the normal sample, let me take that kind of example where we are comparing or try to find out somatic mutations in the cancer. So let's go over again here under the genomics section. So here under mutation variant, parallel analysis of mutation variant data. Let's take this cancer normal mutation analysis. Or I have simple already results as well. So yeah. So this is variant calling cell line where we already computed some of the results as well. So under the variant calling, here again, as this is an example pipeline, so you need not to upload any data. Here we already provided one cancer sample and one normal sample. So we just need to click on start. So with the start, we able to click on able to upload the sample. Then of course, as we know that in the NGS, we need to perform important steps like pre-processing. Particularly here, we are removing any PCR duplicates using PCR clean. Then butyl 2 to align or reads on reference genomes. Of course, here the reference genome is human genome, so HG38 genome. So then with the STRLCA, which is one of the algorithm, particularly for variant calling or small indel detection algorithm, so we can find important mutations. So then by clicking on the OK, we able to calculate this. So apart from that, you will find other options as well. So right now I'm choosing STRLCA algorithm. So then SMETIC snipper, we are using SMETIC snipper, which is again a Bayesian kind of Bayesian based Bayesian approach based method, so which compare cancer and the normal sample, then it determines whether a particular mutation, so it will provide you SMETIC score. Based on that particular score, you can actually understand whether a mutation, higher the score indicate that mutation is more important. So here we are using this particular SMETIC snipper algorithm. So then we can end the pipeline. So now again, once we complete the pipeline, so we will automatically get this output files. So I'm just downloading this zip folder. Meanwhile, it is just downloading. Let me look at if there is any question in the chat box. Okay, so I have already opened one of the this results in the Excel. So these are the SMETIC snipper results. So eventually from this pipeline, although we will obtain the VCF files, if we analyze those VCF files, now let's look at what kind of results here we have. So this is kind of a table where we able to compute the results, final results. We have chromosome position. First, we have a particular chromosome, then chromosome position, then what is the reference allele. So exactly what was the reference gene, reference base or reference nucleotide in our in case of the reference genome. So then we are comparing it with respect to cancer in the normal sample. So we have, this is tumor sample and this is normal sample. Now apart from ATGC, you will find some of other nucleotide here like R, G, Y, but it indicates. So these are the substitution mutations. Substitution means A, of course, A could be addition mutation. So then we also represented, for example, M. M again indicate it is addition mutation, where in addition to A, the C is also getting added. Similarly, you will find W, there is AT, R indicate AZ. So these kind of different positions corresponding exactly what they represent. Now, based on the somatic score, you can understand whether which particular mutation is more important. So let's here apply a filter, then just sort our values, particularly based on somatic snipper, largest to highest. So larger the value of somatic snipper score, that mutation would be more important. So this is how you can analyze and assess these kind of results, particularly from the mutational analysis or mutation variant calling method, methods. Apart from that, there are also the VCF files that we are going to talk about during this upcoming genomics program, where you can learn, like if you have VCF files, how you can assess what are the mutations in a particular gene, so how you can analyze that. And that is what we are going to talk about in our upcoming session, as well as these things in much more detail. So yeah, that's it. If you have any question, I would be happy to answer your question. No, I would like pass this session back to Dr. Raghav. So Dr. Raghav, over to you.
Thank you very much, Dr. Harpreet. Let me find ways to share my screen. Yes. Okay, welcome back, everybody. Thank you, Dr. Harpreet, for an expertful presentation on how the bioinfo platform can be used or leveraged for genomic analysis. So as I was mentioning before the break, the platform, the OmicsLogic platform, is designed to help you learn more about these analysis methods and their applications in various project examples. So the program in particular that we are going to present will include many more expanded overview of the key concepts, principles, and examples of genomic data analysis and mentor support and all the associated credentials to perform these analysis. So let's move on to how are such genomic studies used in real life. Let's move on to the applications of these genomic studies. So what kind of research questions do they answer, and what is the application of such an extensive analysis of genomic data? So we will discuss few examples today and few example applications of genomics, and we will find that the applications of genomic studies can be employed to characterize genomic variants or inherited genetic variants that can cause deadly diseases like sickle cell anemia, and that can cause mutations in oncogenes that may cause cancer, and that can cause variations or mutations in specific gene that might cause or that will cause Huntington's disease, or that will cause several mutations or variations which will eventually enable a pathogen to develop resistance to the drug that is used to kill them. So first, upon receiving the DNA sequence of a sick patient, the first question we generally ask is, what genes are different, what genes are affected? But due to the volume of the sequence and due to the number of variations that one usually finds in sequencing output analysis, this is not a simple and straightforward question to answer. Even though genomics is currently being employed and used for diagnostics in clinical care purposes, many DNA variants are still being discovered and are still being annotated and are still being rigorously analyzed by researchers and by scientists from diverse teams. So for example, in cancer, the DNA variations can help us to determine the genetic risk associated with the oncogenes. But most often, oncologists will look for somatic mutations in tumor suppressor genes, in oncogenes, or in DNA repair genes specifically. Since cancer most often occurs with age, many researchers see the accumulation of mutations in key genomic regions. These accumulated variations will occur as a combination of driver mutations that causes the cancer and passenger mutations that tag along with the driver mutations. And identifying such mutations and variations in these key genomic regions can help us diagnose cancer very early on, or can help us evaluate the risk of the cancer severity, and to help us eventually point us to select appropriate treatment that will slow down incurable metastatic cancer situations. So one example is for a gene in particular P53, tumor protein P53, which is responsible, this protein is responsible for tumor suppression. So it is implicated in many cancer that this gene prevents tumor growth through a process called apoptosis. So some mutations in these genes that are characterized, some mutations in these genes can lead to loss of function, ultimately promoting tumorigenesis by not controlling the replication of cells with the damaged DNA. So this is an example from cancer. Yeah, those are the mutations. And other diseases like cystic fibrosis can also be linked to individual genes, like the CTFR gene, which can affect the function of a channel that is responsible for clearing the mucus from the airways, and hence cystic fibrosis is born. And in the case of Huntington's disease, which there is an inherited condition, there is a genetically inherited condition from parents or from chromosomal combinations, where a specific gene on chromosome 4 has an abnormal number of CAG codons that is repeated. So this HTT gene encodes a very large protein called Huntington protein, and the additional repeats in this gene will change the structure of the protein, resulting in movement disorder and resulting in dementia and resulting in behavioral abnormalities. So in around 2015, the Huntington Consortium published an excellent study of over 1,000 individuals with early and late onset of disease. The genomic analysis and targeted sequences and whole genome sequencing of this data, of this analysis, from this analysis, the researchers discovered important regulatory mechanisms that are different between these two groups, one group which develop early and one group which develop late onset of disease. So this study also identified, okay, let's go back. Yeah, that is the, that is this one. This study also identified important mutations as shown here in chromosome 15 that are to be associated with the slower development of clinical onset. So this research, as well as others, are excellent examples of how genomic studies and extensive genomic studies can help us identify mechanisms of disease progression and help us identify potential therapeutic interventions using the natural mechanisms of disease regulation. So in another example, tuberculosis is a contagious infection that usually attacks lungs. It can also spread to other parts of the body like brain and spine and bone, etc. A type of bacteria that you all know called Mycobacterium tuberculosis causes it. But the severity of this disease is very well known, and it is still one of the leading causes of deaths all over various countries. Today, most of the cases are cured with antibiotics. However, recently, this bacteria has become more and more resistant to single types of antibiotics. It turns out the resistance is a result of mutation on specific genes or specific promoters. So understanding which genes and which promoters that are involved in developing this resistance to antibiotics and the mechanism of this resistance development are very critical and crucial to develop new drugs and to identify a multi-drug resistant tuberculosis infection from which patients can be easily saved. So in another example that relates to current situation, viruses are extremely prone to acquiring mutations because of the number of times that they replicate themselves and the limited machinery that they have in proofreading the replication process. So the Ebola virus, for example, is an RNA virus that causes severe disease with a very high mortality rate. During the outbreak that happened in 2014 and 16, the genomic sequencing efforts has been applied in near real-time track to understand and to model the pandemic spread and to develop a vaccine. As a result, many researchers focus their attention on potential mutations that can have profound effect on vaccine efficacy as well as viral transmission rates. This happened in 2014 and 2016, but we are seeing the a similar track that is unraveling for SARS-CoV-19 virus, the efforts in characterization of different variants, and the efforts in understanding the efficacy of multiple different vaccines that we have in our hand. So in this workshop today, we reviewed the fundamentals of genomics as well as the bioinformatics tools and the applications of such tools to various research, clinical, and industrial challenges. And we demonstrated you how to perform such analysis in T-BioInfo platform. I hope this workshop and such and today and session today will help you in appreciating the concepts, the data, and the analysis method that is used in genomics. To learn each and every, to learn more about each and every one of these steps in detail, please subscribe to the Genomics program that is starting shortly. And I will be happy to take any questions if you have. And please present your questions in the chat room. And that will conclude today's session. Thank you all for participation. I'm waiting for your question. I will pass the stage back on to Sonalika, I think.
Yes, thank you so much, Dr. Raghavendra, for the session. And it was very insightful and nice to learn more about the field and also get some good hands-on. So I see that many of you were not able to access the server bioinfo. So when you log in or like create an account on the learn.omicslogic portal, it takes some time to get your credentials active on the server. So if you were not able to sign up now, just try in a while, maybe after a few hours, and I'll be sharing the recording as well with all of you and try to replicate the steps that were mentioned by the mentors and practice the pipeline. And tomorrow when we meet in the workshop, please come up with the questions, or if you face any of the issues while running the pipeline or you face any difficulty understanding something, then do let us know. And if we have any questions for now, then please let us know in the chat box so that we can assist you with the same. Now, I would like to invite our Business Development Associate, Ms. Parsh Thar, to just take us through the registration steps for the Genomic Data Analysis program that is starting from Jan 18th. So, Sparsh, are you here with me?
Yeah, Sonalika.
Yes,
Is
I
my
can
voice
share the
too
screen
loud?
for you. Yes, we
Yeah,
can hear
sure.
you. Also, we have a question. Naila, you can't export the pipelines, but you can export the results that are obtained by running the pipeline. Yes, Dr. Harpreet has also answered the same. Thank you, ma'am. And Sucheshna, we will be sharing the recording just within few hours when the processing of the recording is over. So you will be able to get the recording in just 4 to 5 hours from now. We have one more question. Okay, I believe that has been answered by Sparsh. So yes, Sparsh, this is the program page that I would like to take you all to, the Genomic Data Analysis program page. The program starts on 18th of January and will conclude on 18th of February. Now, the workshop that we have scheduled for these 2 days, the 11th and 12th of January, is just a glimpse and overview of what all would be covered in the program, and just to get the interested participants started with genomic data analysis, right? So on this program page, Marks and Hardik, we'll just take your questions just in a while. Just give me a few seconds so that I can just go to this program page and then we'll be able to answer your questions. So on this program page, you will be able to find the program overview, and this video will give you an overview about the program and what it is all about. And on the program page itself, you can see that the schedule of each and every session has been mentioned, the date for which the session is scheduled, the topics that would be covered in that particular session, and also along each and every session, you'll find some associated online resources that the program participant is supposed to go through and complete. Now, these associated resources are linked to the lessons on the OmicsLogic Learn portal. So we have tried to design this program in a way that the participants can go through this asynchronous training as well, along with the synchronous live sessions, and get mentor guidance. And also here on the program page itself, you will find all the sessions. And once the session is completed, the recording of the session will be uploaded here under each session itself, so that you can go to the recording at your own pace. So let's say if you're not able to join a particular session, or you were not able to go through the complete live session, you can definitely look at the recording once again after the session is over, or at your own convenience. Now, to register for this particular program, You will just scroll down on the program page itself and you will find 3 levels. Now, to discuss about these 3 levels and to help you out with your enrollment for the program Genomic Data Analysis, I would like to invite Sparsh to please help us understand these 3 different levels and help the participants also understand how they should get started with the program enrollment. Over to you, Sparsh.
So thank you, Sonalika. So I'm Sparsh, Business Development Associate at Pine Biotech, as Sonalika already introduced me. And I'm here to assist you with the enrollment for Genomics Data Analysis program, which is commencing from 18th of this January. So for that, you need to follow 2 steps. One is to register yourself for the Genomics Data Analysis program. And the second step is that you need to opt for you need to opt any of the 3 level of subscription, as you can see on the screen. So the first level is the beginner level, the second level is the intermediate level, and the third is the advanced level. And the fees for the beginner level is $75. The fees for the intermediate level is $105, and the fees for the advanced level is $175. Now, what's the difference between the 3 level of subscription? In the beginner level of subscription, you will be assisted in live group sessions. So you will be connected with our mentors through our Slack channels, and also in a Q&A sessions, which will be conducted weekly. But in the intermediate session, you will be having an opportunity to have one-on-one mentor support, one-on-one mentor guidance. And in the advanced level, you will get an 1 month extra time to do a project under mentor guidance. Now, except these 3 specific resources which you are getting in the 3 level of subscriptions, what all other resources you will be getting is online tutorials, project examples, cloud pipelines, coding sessions, session recordings, and also you will be certified after the completion of the program. And also, I would like to add here that in advanced level of subscription, you will be certified with the completion of the program, and also you will be getting a certificate of excellence. Now, particularly for Indian contacts, we have created a UPI ID so that to make the payment checkout more feasible for them. And to know more about the enrollment process through the UPI ID, so I'm mentioning in the chat box the mail ID where you can ask anything and clear off your queries. So the mail ID is marketing@omicslogic.com. So you can mail us at this particular mail ID if you get stuck in between during your enrollment process. Now, if you click on the select option, Sonalika. Hello, Sonalika, can you do this for me? Yeah. If you'll click on the select option, so it's getting loaded. I think so there's some network issue. Yeah. So here there you will be directed to the window where you need to fill your card details and proceed with the payment checkout. Now, yes, we are also providing everyone with an early bird discount of 20%, and those who are interested, please can leave their email IDs in the chat box, and we'll revert back you through the email through the mail itself. So I hope the enrollment process is quite clear, or if you are having any doubts, you can uh, please ask in the chat box.
Marks and Naila, I think your questions have been clarified, and thank you, Dr. Raghavendran, for answering Hardik's question. So you can definitely go through the coursework and the projects here on the portal and explore these various types of specialization tracks that you would like to explore. So Hardik, this is just for you. If you just like go to projects and you'll be able to here look at a specific project which has used these, which comes under these categories, that is on drought, stress, and resistance in potato. And this is a very nice project example that you can definitely go through and get some hands on. Any other questions do we have? Okay, so if that was it for today's workshop, then we can take the questions tomorrow once you have gone through the concepts and the pipelines as well that Dr. Raghavendran and Dr. Harpreet took us through today. So now we'll be meeting tomorrow as well, 1 hour prior to the scheduled time of today's. And those who have not yet received the invitation link for tomorrow's workshop can please let us know by mailing at the mail ID provided by Sparsh, that is marketing@pine.bio, and we'll be able to send you the invitation. And the workshop tomorrow will also have some exciting sessions. We have a speaker from BioCause, Dr. Sanjeev will be joining us, and he'll be taking us through single cell data analysis. The timing for the workshop tomorrow is 7:30 p.m. IST or 8 a.m. CST. We'll be looking forward to meeting you all tomorrow. Have a great day and thank you for joining today.